Hello everyone, you're watching Blackboard. As in the previous lecture, we discussed about the basics of serology and its applications. Today we will learn about one of the serological assays known as ELISA. The host or body is prone to infection due to foreign infectious pathogens which may be bacteria, virus or fungus. And in order to protect against these pathogens, our body recruits specific antibodies to kill or neutralize these antigens as a protective mechanism. So the presence of infection can be serologically identified either by detecting the presence of antigen or antibody raised against that particular antigen. For identifying these antigens or antibodies, ELISA can be done, abbreviation for enzyme-linked immunosorbent assay. ELISA is the most commonly and routinely used serological-based disease diagnostic method. It was first described in 1971 by Eva Engvall and Peter Perlman. Before the development of ELISA, the only option for conducting an immunoassay was radioimmunoassay, a technique using radioactively labeled antigens and antibodies, and an analyte concentration was detected by measuring radioactivity. ELISA is the colorimetric technique which is used to measure the concentration of various analytes, including antigens, antibodies, and hormones, by a color development reaction. It gives quantitative and qualitative information about the presence of antigen or antibody. It is a wet lab technique because the analytes to be measured is in a liquid state and liquid reagents are used during analysis. Do you know that ELISA was the first screening test used for HIV because of its high sensitivity? It is used to diagnose many viral infections including HPV, HCV, rotavirus, and many other diseases like syphilis, dengue, and malaria. ELISA follows the principle of immunology. It's all about antigen-antibody interactions. According to lock and key model, a specific key fits into the lock. In the same manner, ELISA uses antibodies that are specific for a particular antigen. That's why it's a highly sensitive technique. For ELISA test, we need a 96-well flat-bottom microtiter plate coated with polystyrene. The analyte to be measured, which can be an antigen or antibody, is adsorbed on the plate. Then an enzyme-linked antibody is raised against the analyte that converts colorless substrate into a colored product. The enzyme can be horse-radish peroxidase and the substrate is TMB. The color produced is quantified by a colorimeter that measures the absorbance of specific wavelength, and result is displayed as the absorbance versus concentration graph in the computer screen. The intensity of color corresponds to the amount of analyte in the sample, which means greater the intensity of color, greater the analyte concentration. There are several types of ELISA that differ in the way the analyte is detected. The most common types are direct ELISA, indirect ELISA, sandwich, and competitive ELISA. In case of direct ELISA, as the sample is added into well, antigens from the sample gets immobilized on the plate. Then enzyme conjugated antibody specific to the antigen is added. The wash is given to remove unbound antibodies. Substrate is added and a color reaction takes place. Indirect ELISA is used to detect antibodies against a known antigen. The antigen is already bound to the bottom of microplate well. Then sample is added and specific antibodies bind to these antigens. A secondary antibody conjugated to an enzyme is added, which is specific for the primary antibody and converts the colorless substrate into a reaction product, and the intensity of color is measured. Sandwich ELISA is used to detect antigen of interest. In this, two antibodies specific to different epitopes on the same target antigen are used. The capture antibody is bound to the bottom of microplate well and binds one epitope of the antigen when sample is added. Then a secondary antibody conjugated with the enzyme is added and binds to antigen at a different epitope. Then a secondary antibody conjugated with the enzyme is added and binds to antigen at a different epitope. Competitive ELISA is used to measure small molecules like T3, T4 and progesterone hormones. The result interpretation of competitive ELISA is different than other types because in the previous one we learned that intensity of color produced is directly proportional to the amount of analyte. But in case of competitive ELISA, the intensity of color is inversely proportional to the concentration of analyte in the sample. Incubate the antibody with the antigen to be measured. Now add this antigen-antibody mixture to a microplate well pre-coated with the same analyte. Now add enzyme conjugated antibody, add substrate, and color intensity is measured. The higher concentration of antigen in the sample leads to an increased number of antibodies to bind with them. Consequently, less antibodies are available to bind with the coated antigen and less the color intensity. The materials required to perform an ELISA includes a microwell plate coated with polystyrene containing 96 wells, an 8-channel pipette for easy pipetting, automated microplate washer, they are very efficient with low carryover contamination, incubators for efficient antigen-antibody binding, and a microplate reader. The reagents available in the ELISA kit include dilution buffer, a wash buffer to remove unbound antigens or antibodies, a blocking buffer, that is bovine serum albumin, used to block unoccupied spaces in the well and prevent non-specific binding, enzyme conjugate, a substrate, and a stop solution, that is 0.5 molar H2SO4. As the most commercially available ELISA kits are for sandwich ELISA, so let's have a look at its procedure. Capture antibody is already bound to the ELISA plate wells. Add sample to well so that antigen within sample binds to capture antibody. After incubation, wash the microplate to get unbound material washed away, leaving only the antigen of interest. Now add detection antibody. This enzyme conjugated detection antibody binds to a secondary site on the antigen of interest. Wash microplate so that unbound antibodies are washed away. Now add substrate. Substrate is converted by the enzyme on the detection body, producing a color change. Read the plate. The plate reader detects the color reaction product and outputs optical density values. In the last, calculate result. The amount of antigen in each sample is calculated and analyzed. So that's all about today's lecture. If you liked the video, hit the like button and subscribe my channel. Thank you.